Hi, my name is Jordan McCrae, and I am doing my final project on genetics and linear algebra. I chose to do this subject because I have a past interest in genetics. Um, when I was in high school, I took a couple health science courses, and I just remember genetics was something that stuck out to me, and I thought it was something I wanted to do in the future as a career. And yeah, so I have a lot of background knowledge on probability with genetics and stuff, and lots of vocabulary in this subject. So I just figured why not connect it with linear algebra today. So the topic is how matrices explain inheritance. Here is my glossary. This is not every single word that I use, but these to me were the main ones that stuck out. And throughout the entire presentation, I have tried to bolden every single time I used something from the glossary, but there might have been a couple that I missed. But yeah. Background. What are genes? Genes are segments of DNA that carry the instructions for building proteins. These proteins influence the physical and functional characteristics of an organism. What are alleles? Alleles are different versions of a gene. Example, a gene for flower color may have a red allele and a white allele. How do traits form? Traits result from the combination of alleles inherited from each parent. Each individual receives one allele from the mother and one allele from the father. Together, this pair forms a genotype such as two big A's, a big A and a little a, or two little A's, which expresses a phenotype, the observable trait, which would be something like eye color, hair color. Why this matters for our topic: inheritance patterns follow consistent, predictable rules. Because these rules involve combining information, tracking ratios, and analyzing patterns across generations, they can be described mathematically using vectors, matrices, and linear transformations. Why genetics uses linear algebra? Key ideas are that genetic traits can be represented as vectors. A vector encodes information such as genotype frequencies or allele counts. Trait mixing follows vector addition. Combining parent traits works like adding vectors component-wise, similar to the parallelogram law. Inheritance patterns from linear combinations. Offspring genotypes arise from weighted sums of parental contributions. Population changes across generations can be modeled with matrices. A matrix vector equation can describe how one generation transforms into the next. Linear algebra provides tools to detect structure in genetic data. Concepts like rank, column space, and linearly independent markers help identify which genetic traits are redundant or essential. How genetics uses linear algebra. Why this matters in biology: helps predict genotype and phenotype ratios, models long-term allele frequency behavior, explains equilibrium states using eigenvalues and eigenvectors, allows classification and reconstruction of genetic patterns. And here's just a quick Google image of a Punnett square and how the parents' alleles affect the outcome of the children and, the probability and stuff. Representing genetic traits using vectors. How linear algebra describes genotypes in populations. A genotype can be written as a vector. Each coordinate represents alleles or genotype frequencies. And here's an example for Mendelian genotypes. Any population is a linear combination of these vectors. All genetic mixtures lie in the span of these basis vectors, meaning they form a subspace of R3. Why this is useful: allows us to quantify genetic distributions, sets up population models using matrices, and shows how traits combine through linear algebra, coefficients, and vector addition. Punnett squares as matrices. Punnett squares are matrix operations. In genetics, a Punnett square combines allele contributions from two parents. In linear algebra, this same process is represented through matrix multiplication. Each parent's allele probabilities are written as vectors, and multiplying these vectors produces all possible offspring outcomes. Because matrix multiplication is associative, we can extend this method to track genetic patterns across many generations with no change in results. Viewing Punnett squares as matrices allows us to treat inheritance as a linear transformation, making predictions more structured and mathematically precise. And here is an example. I'll give you a second to look at that. Or you can just pause the video. I forgot you can pause. Why this matters: saves time when analyzing large genetic systems, lets us model multiple generations using repeated matrix multiplication, and connects genetics directly to linear algebra concepts like matrix multiplication, associativity, and linear combinations of probability. Modeling populations with matrix transformations, how matrices predict genetic change across generations. In population genetics, we often track how genotype frequencies evolve over time. Linear algebra models this process using a transition matrix, where each entry represents the probability that one genotype transforms to another in the next generation. When we multiply this matrix by a population vector, we obtain the genotype distribution for the next generation. This framework treats inheritance as a linear transformation, allowing us to analyze long-term trend, stability, and equilibrium behavior using structured mathematical tools. What this means biologically: each generation is obtained through matrix multiplication. The matrix describes how alleles combine, mutate, or survive. Patterns seen after many generations reflect the structure of A, helps biologists predict long-term allele frequencies. Why this is useful: it works even for large or complex genetic systems. It matches real-world inheritance patterns and leads into important ideas like eigenvalues, eigenvectors, and equilibrium distributions. Eigenvalues and genetic equilibrium. How eigenvectors describe stable genetic distributions. When we repeatedly apply a transition matrix to a population vector, the distribution of genotypes often settles into a stable pattern that no longer changes from one generation to the next. In linear algebra, this stable pattern is an eigenvector of the transition matrix. If a population vector X satisfies the equation A times X equals lambda times X, then X lies along an eigendirection of the matrix A. In genetics, this eigenvector represents an equilibrium distribution of alleles or genotypes, and the eigenvalue lambda indicates whether the population grows, shrinks, or stays consistent or constant in that direction. A is a transition matrix, V is the equilibrium population vector, and lambda is the eigenvalue, often one in genetic equilibrium models. Eigenvalues in genetic equilibrium. Biological meaning: V stays proportional to itself after reproduction. The population settles into the same genotype ratios each generation. Lambda being 1 means the system stabilizes, no long-term growth or decay. Lambda being less than 1 means that pattern dies out, and lambda being greater than 1 means that pattern expands over time. Why eigenvectors matter in genetics: they predict allele frequencies many generations into the future, identify stable genetic distributions, analyze effects of mutation or selection, and provide insight into long-term evolutionary trends. Solving genetic constraints using augmented matrices. Using linear systems to analyze genetic relationships. In many genetic problems, genotype or allele frequencies must satisfy certain linear conditions. For example, that probabilities sum to 1 or that a phenotype is produced by a combination of genotypes. These relationships can be written as a system of linear equations and solved using an augmented matrix. Applying Gaussian elimination allows us to determine whether the system is consistent, whether multiple genetic distributions satisfy the same phenotype, or how many degrees of freedom exist in the model. This links biological constraints directly to algebraic structure. What Gaussian elimination reveals: if the system is consistent, the genetic model is possible. Missing pivots indicate free variables, meaning multiple genetic distributions fit the phenotype. One pivot per variable gives one unique solution. Fewer pivots equal infinite solutions, biological flexibility. Why this matters in genetics: helps test whether proposed genotype ratios are feasible, allows reconstruction of missing genetic information, shows how biological constraints shape the solution space, and connects genotype modeling to concepts like pivot positions, row echelon form, and free variables. Free variables and biological degrees of freedom. When solving genetic systems using augmented matrices, we sometimes find that not every variable receives a pivot during Gaussian elimination. These leftover variables are called free variables, and they indicate that there are infinitely many solutions that satisfy the same biological conditions. In genetics, this means multiple genotype distributions can produce the same phenotype or satisfy the same probability constraints. Free variable represents degrees of freedom in the biological system, showing that the data allows several possible genetic explanations rather than one fixed pattern. Here is an example. Here, t is a free variable. This means every choice of T gives a valid genetic distribution as long as probabilities remain between 0 and 1. Free variables are multiple genetic distributions. Free variables equal multiple genetic distributions fit the same constraints. The solution set forms a line or plane in genetics genotype space. Biology rarely forces a single exact distribution. There is often a range of possibilities. These degrees of freedom come from the structure of the linear system, not randomness. Rank in genetic data, identifying independent traits. In genetics, we often work with data matrices and record measurements of multiple traits across individuals. The rank of this matrix tells us how many traits provide genuinely independent information. If the rank is smaller than the number of recorded traits, then some traits are linear combinations of others and do not provide new genetic insight. Rank therefore helps biologists detect redundancy, understand relationships between traits, and determine whether the collated data spans the full trait space or lies in a lower dimensional subspace. Biological interpretation is that only one independent genetic trait is being measured. The other traits are scaled versions, linear combinations of the first. The true genetic variation lies on a one-dimensional subspace. Rank exposes how many traits are truly independent in the population. Why rank matters. Detects redundant or dependent genetic markers, helps determine which traits carry unique information, identifies whether genetic data can reconstruct missing values, and connects empirical data to concepts such as column space and span. How linear independence identifies useful genetic markers. In genetics, we often measure multiple markers to determine whether they provide unique information about inheritance patterns. Two or more trait vectors are linearly independent if none of them can be written as a linear combination of the others. When traits are independent, they each contribute new biological information. But if a trait is dependent, meaning it can be transformed from a combination of others, it is redundant. Linear independence therefore reveals whether each marker truly expands the genetic trait space or whether some measurements can be removed without losing information. If any non-zero combination equals a zero vector, the traits are dependent. Genetic interpretation. Independent markers represent distinct biological information. Dependent markers measure overlapping or redundant traits. Independence ensures traits span a higher dimensional genetic space. Dependency collapses trait variation onto a lower dimensional subspace. Why this matters in biology. Helps eliminate unnecessary genetic markers, improves accuracy in genetic modeling, and ensures data analysis uses genuinely informative traits. Supports better predictions of inheritance patterns. DNA as a linear code, the Hill cipher analogy. How genetic coding represents or resembles a linear algebra cipher. Although biology and cryptography seem unrelated, DNA sequences behave much like messages encoded with matrix multiplication. The Hill cipher, a classical encryption method, converts groups of letters into vectors and multiplies them by multiplication. By an invertible matrix to provide encoded outputs, Similarly, in genetics, DNA bases can be represented numerically and transformed through linear operations such as mutations, replications, or protein coding processes. Thinking of DNA this way helps illustrate how biological information is preserved or altered through invertible matrices, linear transformations, and vector representations. Why this relates to genetics. DNA bases can be represented as numerical vectors. Replication and transcription transform sequences in structured ways. Mutations can be interpreted as shifts under linear transformations. Decoding genetic information resembles applying an inverse matrix. In the biological interpretation, genetic information is preserved when the transformation is invertible. Some mutations correspond to transforming a vector outside the expected subspace. Patterns in codons resemble structured matrix operations on triplet vectors. Conclusion. How linear algebra supports genetics. Bringing genetics and linear algebra together. Linear algebra provides a powerful framework for understanding how genetic traits behave, combine, and evolve across generations. By representing genotypes as vectors and using matrices to model inheritance, we gain a structured way to analyze population changes, identify stable genetic patterns, and detect dependencies among traits. Concepts such as gene vectors, rank, free variables, and linear combinations reveal how biological systems behave in both predictable and flexible ways. Together, these tools show that genetics is deeply connected to the mathematical structure of vector spaces and matrix transformations. Key takeaways are genotypes and populations can be represented